When using the Incucyte SX1, the user pre-selects the required objective before scanning. Should the objective need to be changed, this can be performed by going to the Device tab, clicking Administer, and in the Configuration menu selecting the Change the Active Objective. Please note that changing the active objective will cancel all upcoming scans. Further details can be found in the Incucyte manual on page 171, Appendix B, Changing the Incucyte SX1 Objective. To start scanning a vessel on the Incucyte, click Schedule to acquire. This will open up the acquisition window where the current vessels scheduled to be scanned are displayed, along with the timeline showing when these scans will occur. To set up a new vessel, click on the blue plus icon on the left side of the screen that says Launch Add Vessel. At various points in the Incucyte software, pop-ups will appear giving the user additional information about different elements within the graphical user interface. This will open up the guided interface containing 11 steps where the user can check the progression at the bottom of the screen. Initially, there are two options available to set up a scan. Scan on schedule, which creates a user-defined repeating schedule, or Scan Once Now, which immediately initiates a single one-off scan. Double-click on the scan type or click Next to proceed to the Create or Restore Vessel page. Now the user has a choice to create a new vessel to scan, copy your current vessel that's currently scheduled, or copy the settings of a previously scanned vessel. There is also the option to restore a previously scanned vessel for additional scanning. The new scans are then added to the existing scans in one experimental file. For now, we're going to choose Create New Vessel. In the scan type page, the relevant scanning mode depending on the assay and application is chosen. Different scanning modes may lead to pre-selection of objectives and tissue culture plastic specific to that scanning mode. The scan types Standard and Image Lock are always available and functional. Whole well and dilution cloning are available with the purchase and selection of the 4X objective. The scratch wound, chemotaxis, and spheroid scan types are enabled upon purchase of the applicable software module and selection of the relevant objective. Most assays, such as proliferation, cell death, phagocytosis, and 2D immune cell killing, are run using the standard scan type. Standard scanning also allows the selection of the cell-by-cell scanning options. Should your module be enabled and the 10x or 20x objective be selected. In the Scan Settings page, the desired image channels are selected. The objective is selected in the Device tab, as discussed earlier, and the same objective is used for all loaded vessels. The fluorescence acquisition time, which is comprised of a series of exposures, is the length of time that the cells are excited. The acquisition time options are set to default values of 300 milliseconds for green and 400 milliseconds for red fluorescence. We have determined that these default times are the optimal exposures when scanning across a wide variety of fluorescent reporters using the Incucyte. However, should it be necessary, it is possible to modify these values. For example, in the case of fluorescent signal saturation when using a bright fluorophore. The vessel selection page lists all vessel types that the Incucyte system can support. Should a scanning mode other than standard be selected, then only those vessels which are applicable for that scanning mode and objective will be displayed. Users can search for vessels in several ways, for example by manufacturer, catalog number, or number of wells. In the vessel location page, users can select the position in the Incucyte where their vessel is located. Recommended best practice is to physically load the vessel into the Incucyte gantry before beginning scheduling. For the Incucyte SX1, only the front tray location is available. Should the selected location already contain a vessel, then a warning message and table appear with the option to remove that vessel from the schedule. The scan pattern page displays a vessel overview, which allows the user to select the locations of wells within the vessel to be scanned. When scanning microplates, it's very important to not select wells which do not contain cells, as this can interfere with the Incucyte's autofocusing. Once locations are selected, holding Alt before clicking and dragging will deselect multiple locations. Next, the user can specify the number of images that are to be acquired per well. Generally, it is recommended to acquire 2 images per well for adherent cell types or homogeneously distributed cells, and 4 images per well for suspension cell types, in a 96 well plate with the 10x objective using the standard scanning mode. The estimated scan duration, based on the chosen scan settings, is displayed at the bottom of the page. On the vessel notebook page, details about the vessel can be added, including the vessel name, the cell type, and the passage number. Clear naming is important to allow vessels to be searchable following scanning. It is also possible to create or import a plate map for the vessel. This allows the Incucyte to group replicate wells when exporting data and acts as an experimental record. Please refer to the How to Create a Plate Map video tutorial to learn how to create a plate map. Notes can also be added to the notebook should they be needed. Once assays and analyses have been run multiple times and are established, it is possible to launch analysis in conjunction with image acquisition. The appropriate analysis type is selected, allowing the selection of the desired analysis definition. Spectral mixing values, having been determined previously, are then inputted along with any notes. If the assay has not been performed previously, then it is recommended to defer the analysis until later and run it post-acquisition. On the scan schedule page, the user specifies the schedule for the vessel. Initially, there are 3 options to add to a schedule. The first user can create a new schedule with defined scan intervals, for example, every 2 hours, independent of the existing schedule. Create a new schedule with advanced scheduling options. This can be opened by right-clicking the timeline and selecting Set Scan Interval. This allows users to define a specific start time, for example, 11 a.m., the scan interval, for example, every hour, and the window of scanning during each 24-hour interval. In this example, choosing a total of 12 hours means that the vessel will be scanned every hour until 11 p.m. However, it is important to note that the schedule repeats daily. So in this example, scanning will recommence at 11 a.m. the next morning for an additional 12 hours. Leaving the window of scanning for a total of 24 hours will fill the timeline. Individual scans can also be added to the timeline by clicking. This scheduling option is also available in the schedule overview by double-clicking the timeline. A user can also reserve a tray location and defer scanning until later, with vessel scheduling being performed in the schedule overview by double-clicking the timeline. Following schedule selection, the duration of the experiment can also be defined. Scan indefinitely will continue scanning until the user manually stops the experiment and is recommended for new experiments. The user can also define a specific endpoint, either a number of days or hours after the experiment commenced or a specific date and time. Following scheduling, the summary screen displays the selected scheduling options. Here the user can click on each page name to automatically go to that page to make any changes. Once the user is satisfied with the schedule, clicking Add to Schedule adds that vessel to the timeline. The vessel and its schedule are now visible to all users. To add a subsequent vessel, the user repeats the previous steps and is obliged to group the new vessel with the existing vessel. Each vessel will be scanned sequentially at each defined time point.